So in this second video with regards to the module 5.15, in particular focusing upon the structure of the muscles, sliding filament theory, and in this particular video we're also going to have a look at the neuromuscular junction and how that is working. I just wanted to do a quick recap upon the structure of the muscle that we looked at last time, and also the different filaments that are making up the myofibril, which is one of the organelles of the myocyte, also known as a muscle fibre. So if we can just look at the picture which is on screen at the moment, very super speedy recap for you here. We've got the main bicep muscle. That muscle is made up of fascicles, and each fascicle is made up of muscle fibres, and the muscle fibre is what is the actual muscle cell, and a muscle cell is referred to as a myocyte, and as we can see here, it is a multinucleated cell. The muscle cell is surrounded by a sarcolemma. Now the sarcolemma is the muscle cell's cell membrane, but in this case we refer to this as its sarcolemma. And throughout the myocyte we can see a number of tubular organelles referred to as myofibrils. Myofibrils are made up of both actin and myosin fibres, and we can see sections within that referred to as sarcomeres, and those are denoted by the Z lines at each end of the sarcomere, and we're looking at about 2.5 micrometers in length for each of those sarcomeres. The actin fibres are the thin of the fibres, and here on screen at the moment those are in the black coloration, and then we have got the thick myosin fibres which are denoted in blue. Now if we move across to the micrograph, we can see these have been articulated here in the last video, and we looked at the Z line, the distance between those lines. We noted that the M line was the part which was going straight down the centre of our sarcomere, and at this point we would only have myosin, and that zone which is denoted by myosin only is referred to as the H zone. Our zone which is responsible for contraction is the A band, and we can see here that we have an overlap of the actin and the myosin, and we can see the myosin heads here quite in detail, and we will look at that in more detail later on. And then we have the I band, which is the area which is only actin. Now the area which is only actin is also referred to as the light zone, and the area in which is dark that we can see in the micrograph is referred to as the dark zone, and we can see that is because of the presence of the thick myosin fibres here. Okay, so what I have drawn for you here is a neuromuscular junction. So we can see at the base of this we have got the sarcolemma, and we spoke earlier about this being the membrane of your muscle cell, so your myocyte, and we can see on your sarcolemma we have got some receptors for acetylcholine. And one thing to note here is that this membrane is highly folded, and actually within the membrane we have very deep folds, so almost tube-like projections within there that fold in, and these are referred to as the T tubules, and those are going to become important when we look at the transmission and the depolarization of your muscle cell, and therefore your stimulation of your contraction to take place. So if we turn our attention now to the neuron, which is bringing the action potential, so we will see the action potential that is going to arrive here at the muscle cell and arrive and then be transmitted across to the sarcolemma of the muscle cell. What we need to recognise here is that this is actually a branch off of the motor neuron. So a motor neuron, as it moves towards the muscle itself, is going to branch into a number of motor units, and at the end of these we have a motor end plate, and that motor end plate will then meet at the sarcolemma, and at the point where it meets the sarcolemma and we see all of these grooves, we refer to this as the junctional groove, and this is the highly folded part of the sarcolemma where it meets with the motor end plate. So if we look at what is happening at the motor end plate, we can see we've got our action potential arriving at this motor end plate from down that motor neuron. And as that arrives, we will see a wave of depolarization taking place. And due to that depolarization of the motor end plate of that motor neuron, we will then see the movement in through these channels of our calcium ions. So we're going to see our Ca2 plus ions moving into the motor neuron as a result of that. Now that Ca2 plus is going to bind with and activate the movement of your vesicles to the membrane of your neuron, and then we will see the release of the acetylcholine into that that space between the junctional groove and the motor neuron itself. And we can see here that we've got some ligand-gated receptors, and that those ligand-gated receptors are specific for your acetylcholine. And when that arrives at the sarcolemma, that's going to result in binding with and then the depolarization of your sarcolemma. So in the same way as you would see if we were moving from a presynaptic to a postsynaptic neuron, and we saw the binding of your acetylcholine with these ligand-gated receptors, we would see them binding. So we would need our 2 acetylcholine molecules to bind with that, and these would then open, and we would see our Na plus ions start to move into our what we would normally be looking at as a neuron, but in this case we're now moving into the sarcolemma. So we can see the sodium is going to depolarize the membrane of the sarcolemma. Reminder that the sarcolemma is surrounding your muscle cell. As we mentioned before, we have these deep grooves that are referred to as T-tubules, and what we can see here is we can see an influx of sodium which will move down those T-tubules, and this is going to flood the sarcoplasmic reticulum. Now the flooding of sodium into the sarcoplasmic reticulum is then going to cause the release and trigger the release of calcium ions from that sarcoplasmic reticulum. And remember, the sarcoplasmic reticulum is engulfing, or is surrounding, I should say, the myofibrils, these organelles sitting within the myocyte. Okay, so just to recap very quickly, we have our action potential which is coming down our motor neuron, and that motor neuron branches into a number of motor units. Those motor units end at a motor end plate, and that is at a junctional groove, which is a highly folded part of your sarcolemma. Sarcolemma being the membrane that surrounds your myocyte, your muscle cell. And that those highly folded sections have deep grooves within them, referred to as T-tubules. When the action potential arrives and causes the depolarization of your motor end plate, we see the influx of the calcium ions, and those calcium 2 plus ions are going to activate your vesicles with your acetylcholine to move to the neuron's membrane, and then be released into the neuromuscular space of the neuromuscular junction. That is then going to activate your ligand-gated acetylcholine receptors, allowing the influx of sodium ions, and that is going to depolarize your sarcolemma, and that's then going to cause the influx of your sodium ions down the T-tubules, which will then depolarize your sarcoplasmic reticulum, and the depolarization of the sarcoplasmic reticulum then results in the release of calcium ions from that sarcoplasmic reticulum, and that's going to be of great importance when it comes to the movement of our muscles and the contractions of our muscles, which we will look at in the next clip.